Hello, everyone. Welcome to the fifth workshop on excitons by the initiative of Theoretical Sciences of the City University of New York. And my name is Sergei Zhang, and along with Chris Bardin at UC Riverside, we have been organizing a series of workshops on excitons in lieu of Telluride workshop. And this has been going on great. And today we have four exciting speakers who are going to talk about quantum dynamics of excitons and Exciton-Light interactions. And there are three theoretical and computational scientists, and also one spectroscopist. And they all use state-of-the-art methods and techniques to explore quantum dynamics of excitons. Without further ado, let me introduce you our first speaker, Irene Burghardt from Goethe University, Frankfurt. Irene, you can show your presentation and get started.
All right, so it's a very great pleasure to be here and start this theory session with a presentation on quantum dynamics of exciton transport and semiconducting polymer chains, coherent or diffusive. So our objective is first principles description of excitonic processes. So we want to look at exciton transport and also exciton dissociation based on real-time quantum propagation. And in fact, we're using exact quantum dynamics in the sense that we have in particular the multi-configuration time-dependent hereditary MCTDH method, which I will give a few more details later on. And so the idea is to fully capture the coherent aspects of the dynamics and also provide benchmarks for more approximate trajectory-based methods. And this is actually a picture of a simulation result. This is actually an exciton that is moving in a fluctuating landscape due to torsional dynamics. Right, so I will first give a few details about the simulation approach, and then we go to the main topic, which is about intra-chain exciton migration guided by conformational dynamics. And the final part, if time permits, is about an outlook towards interchain exciton transport. Right, so here's our general perspective. So we would like to construct lattice type Hamiltonians with input from supramolecular electronic structure calculations. So we start with calculations on oligomers, and we transpose that information, like technically by using diabatization and so on, to this lattice type representation. For example, for a J-type system or an H-aggregate type system, like for a typical conjugated chain or these stacked aggregates. And from the electronic structure side, we get a lot of information, including actually the character of the excitonic state. So we can use a transition density analysis to generate these electron hole maps that give information on like whether we have a pure Frenkel state, more or less, or we have like admixtures of charge transfer excitons, like on the right-hand side, where we have this H-type aggregate that is, however, significantly modified because of the charge transfer admixture. And we also get information on the conformational dependence. So here is an electron hole map as a function of the torsional dynamics, and we can actually see that a conjugation break occurs at some point. So the excitonic density actually goes to 0 at the center of the lattice here because of a conjugation break. Okay, so with this, the idea is to pick up full electronic structure information and map this into an excitonic or electron hole Hamiltonian. And the next step is to include the vibrations. So Electron-Phonon coupling is a key aspect, and we have several ways of doing this. So one way is to use the so-called linear vibronic coupling model, where we use a shifted harmonic oscillator model. And we have the spectral densities that carry information about the shift in equilibrium geometries between the relevant states. Now, this harmonic model works in many cases, especially if we have more or less rigid geometries. On the other hand, if we have soft modes and very flexible modes like torsions, for example, it is important to include unharmonicities. And for that purpose, we're using a different procedure. In this case, it's a mapping procedure from oligomer potential energy surfaces to like this lattice Hamiltonian in a monomer basis. And this is what I'm going to explain in more detail later on. Now, once we have all this, like a lattice Hamiltonian, including the vibronic information, we're going to do quantum dynamics. And as I already mentioned, we're using MCTDH, so multi-configuration time-dependent tertiary. That's a very powerful technique that actually represents correlated time-dependent wave functions as sums over products. And this actually works so well because we have like basis functions here that are time-dependent. And the procedure is variational. So we're actually going to get the best possible way of adapting these time-dependent so-called single particle functions to the dynamics. And this is why that method actually works for like typically up to 100 modes as compared with very few, like up to 5 modes or so with a standard quantum propagation technique. Now, these are the equations of motion, so we have to pay a price in the sense that the equations look complicated. So we have these variational equations for the single particle functions that actually involve multi-configurational mean field Hamiltonians, and everything is time dependent. So this is actually kind of a challenging way of doing quantum dynamics, but it's pretty accurate. And in the simulations that I'm going to show today, we're actually using a more sophisticated variant, a so-called multi-layer MCTDH technique. This is going to take us towards more than 100 states of vibrational modes. And how the idea is actually that we do sort of a repeated MCTDH scheme. So we take each of these single particle functions and we again develop this in terms of this multi-configurational sum over products expansion. So we're sort of doing repeatedly MCTDH and we generate these high dimensional trees. And that is the key to go to a really large number of modes, as I will show later on. Okay, so this is essentially the setup, and the last detail I'd like to mention is that we can combine this with thermalized dynamics. So we want to have non-equilibrium evolution under the influence of temperature. And in the example I'm going to show, we're actually going to do stochastic MCTDH propagation involving like thermostats, molecular thermostats that are described as a so-called Hartree bath. So in this case, there is actually a product assumption here. That is that we have like these thermostats that are attached to individual modes in the MCTDH calculation, and that is a way to get thermal driving of the dynamics. Right. So with all of this, the idea is really to do quantum dynamics as accurate as possible and capture quantum coherence correctly. So in this picture that most of you probably know, so we actually show the key factor that influences electronic coherence. So we have these wave packets that evolve on potential energy surfaces. And when we calculate the electronic coherence, the key issue is about the overlap of these wave packets. And that is the information that we get accurately if we do, for example, MCTDH. And that is exactly what we don't get accurately if we do like simple trajectory-based techniques. And so that's a strong reason for actually trying to do like more or less sophisticated quantum dynamics, even for large systems. All right, so with that, we're going to move to the main application that I'm going to talk about, which is exciton migration guided by conformational dynamics. So we want to know how excitons migrate in typical polymer chains. And so we want to do this in a sort of unbiased representation. And this is why we actually go to a monomer-based picture. Because the partitioning of the system is sort of not predetermined. It's going to be flexible. And we want to know how does the excitonic density actually evolve as a function, for example, of the torsional modes. So we want to have like an ab initio parameterized lattice Hamiltonian of the type I've showed before. So we have this lattice basis, and we want to cast it in the monomer representation to get an unbiased picture. Okay, and the basic Hamiltonian we have in mind is the Frenkel-Holstein Hamiltonian. So that's the classic Hamiltonian one would use to describe exciton migration. So we have side-to-side nearest neighbor couplings, and we also have these side local, usually high-frequency modes that are attached to each side. But we also want to look at these low frequency modes. And we want to have like the unharmonic contribution of the low frequency modes. So we're looking for a significant improvement over the Frenkel-Holstein Hamiltonian. And that is why we use this procedure that I mentioned before about like mapping potential energy surfaces that are computed in an ab initio fashion onto a Frenkel-Holstein type model. So we do calculations for oligomers, and actually in this case, it's like high level ADC2 calculations. So we get the excited states, and so these are like adiabatic eigenvalues. And now we use a mapping procedure that actually uses like the solution to an inverse eigenvalue problem and maps these eigenvalues to a nearest neighbor coupled Hamiltonian, which is going to be like a Frenkel-Holstein Hamiltonian, but much more complete. It's going to carry actually the full electronic structure information for various modes, like torsional modes, high frequency modes, and so on. And just to explain about this inverse eigenvalue mapping, it's actually a very simple idea. So think of a Huckel type matrix. Yeah, so we have analytical solutions for the Huckel problem. The lambdas are the Huckel eigenvalues. And if we want to get the diagonal and off-diagonal values of the Huckel matrix, yeah, we just solve for these in terms of the adiabatic eigenvalues. Now, this is exactly what we do, but it's going to be just much more complicated because we don't have an ideal Huckel matrix, but we have sort of this tri-diagonal matrix that depends on a lot of modes. So it is more complicated, but it can actually be done like partially analytically, and that's what we are using here. Okay, so the result is an ab initio Frenkel-Holstein Hamiltonian. So we have this nearest neighbor coupled type form of the Hamiltonian. And now everything is kind of more detailed. So we have the kinetic energy in terms of curvilinear coordinates. We have side energies that are actually functions of all the coordinates that we've selected. And we have the excitonic coupling. There's also a function like of the torsional modes. And the excitonic couplings are really large, of the order of 1 electron volt, because we are in the monomer-based representation. Okay, and we're doing this for like a chosen subset of modes, torsional modes, and various high frequency modes, like intra-monomer and inter-monomer high frequency modes. And we're constructing this high dimensional potential surface in the shape of a generalized Frenkel-Holstein Hamiltonian. And like for the torsions, it is very important that the excited state potential is actually very stiff, and the torsions are going to planarize in the excited state. So this is what we're going to see in the dynamics. Now, this is dynamics at 0K with just one active torsion to start with, multi-layer MCTDH. For a 20-side system, and we imagine that the exciton is first like confined, let's say to the left of this torsional defect, and we want to see how things change in the course of the dynamics. This is the first 150 femtoseconds, and you see that nothing much happens. So the exciton is like confined on the left-hand side of this defect, and the torsion is slower, so it hasn't moved yet. But we see that the high frequency modes actually adapt to the position of the exciton. So in fact, we have an exciton polaron. Now the exciton is like a trapped species, and the high frequency modes adapt to the position of the exciton. So we have a significant displacement of the high frequency modes. And we also see that there is this immediate decay of the transition Doppler autocorrelation function that actually relates to the experimental anisotropy decay. Now, on a longer time scale, we see that something interesting is happening. So the torsion actually planarizes as expected, and the exciton is following the torsion more or less adiabatically, while the high frequency modes follow the exciton. And we also see that if we switch off one of the high frequency modes, actually the one that is most strongly coupled, that the exciton becomes much more loose, much more spatially extended. So the extent to which we have like this compact exciton species depends a lot on the high frequency modes. And so the exciton has moved to the center of the lattice following the torsion. Why is this? Now, we can see this immediately from the electronic structure. These are the electron hole plots that we showed before as a function of the torsion. So when the torsion is twisted, we kind of see this defect structure. So we see a conjugation break, and the torsion is relaxed. So we go to this local exciton ground state that is nodeless and that extends over the full lattice. So in fact, in the simulation, we started with a linear combination of S1 and S2 of the adiabatic state. So we produce like a partially localized initial exciton, and finally it's going to relax to this local exciton ground state on the extended lattice. So this is what you can see here, right? And so it's like a 2 timescale process where we have the shortest timescale where the trapping is actually happening. So the high frequency modes are going to adapt to the position of the exciton. And then we have like this trapped species, it's polaron exciton, that is going to more or less adiabatically follow the torsion. Okay, and now we're making it more interesting. We go to thermalized dynamics. So now all torsions are active. We have this 20-side system, all torsions active, and we actually have 250 modes here. And you see that the exciton is thermally driven across the lattice. And in a sense, kind of, we see that the exciton is sort of finding a minimum energy path in this fluctuating landscape of torsional motions. And that is actually the data that I showed before initially in this more fancy picture, right? So it's like really an adiabatic dynamics in this fluctuating landscape. And all the way through, the high frequency modes are just following the exciton. Now, these are 2 realizations from our stochastic propagation scheme. And technically, we are doing this multi-layer MCTDH. We have a 7-layer tree, and we have this stochastic Hartree bath. Now, from these simulations, we can extract diffusion coefficients. We can actually compute MSDs, yeah? So mean squared displacements from the simulations, and I'm showing this here actually for 2 realizations. So without periodic boundaries, these are the dashed lines, and with periodic boundaries, these are the full lines, and for different temperatures. So the periodic boundary conditions actually accelerate the dynamics, right? These 20-side systems are too much confined by itself. And then we see that actually the MSDs increase systematically with temperature. And this is actually what is seen in experiment with the exciton transport. We also see that there is an initial ballistic type transient phase that is followed by a normal diffusion regime, so more or less linear. Now, since the dynamics is kind of approximately adiabatic, we sort of, started doing Ehrenfest calculations for comparison. And down here, you actually see Ehrenfest MSD results for 50 picoseconds on a much longer time scale. Yeah. And we sort of followed this up in this particular case. So we thought Ehrenfest could have a chance if we sort of, tweak the method a little bit, so to prevent that we populate higher states and so on. And in fact, it doesn't work too badly. So on the right-hand side is Ehrenfest for the same noise realization as multi-layer MCTDH on the left-hand side. So apart from the fact that we have this spurious oscillations here, so these high frequency oscillations that aren't quite right, actually it isn't too bad. And these are actually also Ehrenfest results, and we see that the adiabatic population, so this is S1 and S2, yeah, are not too badly reproduced, and the inverse participation ratios are not too bad, but it's just extremely noisy because there is actually a high frequency noise due to the S1 and S2 gap. And also we're doing like a systematic damping on the S1 state to prevent like all these problems with Ehrenfest due to the lack of detailed balance and so on. So the idea was to actually go to longer time scales and try to look at the effect of static disorder that we didn't have in the multi-layer MCTDH. And so here are results for actually different values of the static disorder coefficients, like up to 65 milli-eV, which is so representative of P3HT. And you can see that the diffusion coefficient actually goes down as expected. And in the end, as a function of temperature, we get a profile that is actually not too different from experiments. So all this makes sense. Another important observation is that if you look at one realization here, so due to the static disorder, we sort of get like Anderson localization, yeah? And so you see this actually in terms of these hopping type features that are now present in the Ehrenfest dynamics. Now I see that I should actually stop very soon from now. Let me just say a few more things very briefly. So we looked at other systems. So all this was actually oligothiophene. Now we also looked at oligophenol and vinylins. We see that the dynamics is very similar, but actually a bit more non-adiabatic. And we see that besides this dominantly adiabatic transport, we also see effects like resonant interactions between excitons. So this is sort of a different mechanism that can come in for different systems. And actually in the OPVs, we see that the exciton is much more loosely bound as compared with the oligothiophene. Okay, so to wrap this up, we have exciton polarons that are driven by these torsional fluctuations. We have a coherent dynamics that is not band-like, yeah? And essentially we have this picture of excitons surfing along conjugated polymer chains. So this is by this paper of Breda and Silby in 2009. Now, very last excursion to what's going to follow. So interchain transport is very interesting, of course. So recently there have been several examples of extremely fast exciton diffusion in interchain systems. So we started to look at this. So this is the same system that I showed at the beginning. This is an H aggregate actually with this admixture of charge transfer excitons. And so these systems are like on the one hand simpler, on the other hand more complicated than the interchain dynamics. So simpler because these are sort of more rigid, but more complicated because now we have all the photochemistry. So we actually excite states, yeah, at the upper end of the Frenkel manifold. So we get internal conversion, we get de-excitation, possibly conical intersections, and so on. So the photochemistry is going to be more complicated. But so we looked at this at zero temperature, and we actually can see these extended excitonic species, and we can see the effect of electron-phonon coupling as observed in 2D spectroscopy, actually from the Leonel group. So take-home message: coherent effects are important. We think that these ab initio-based lattice Hamiltonians are actually useful. Because they carry the right physics and that can be complementary to other fly dynamics. And importantly, they are compatible with full quantum propagation. And that we now have these multi-configurational methods that are able to reach large system sizes and that give very accurate information about coherence. And so about the exciton dynamics, so the transport is coherent and diffusive, right? So coherent transport, and essentially we observe a normal diffusion regime in our calculations. Right, so essentially exciton polar in motion driven by soft torsional modes, and that should be representative of various other systems. Okay, so thanks to everybody who participated. This is our group at Frankfurt. Thank you very much for your attention.
Great talk, Irene. Let me just get right to some of the questions. The first one is, how did you do your temperature dependence? Is the temperature incorporated through initial conditions or through a thermostating approach? And if it's a thermostat, how do the thermostat dynamics influence the mean square displacement dynamics and things like that?
So, so this is actually a picture of the initial condition. So, so it's like a stochastic wave function setup, in this multi-configurational framework. And in this particular case, we verified that actually the heart rate approximation for the external bath, like for the thermostat, is good enough. This is because these are low frequency rates. So actually each torsion has its own thermostat. And we're doing essentially Wigner function sampling for the bath distribution. So we have a set of like 20, between 10 and 20 explicit bath modes that are attached to each torsion. And so these are equilibrated and then drive the torsional dynamics.
Okay, next question. Any chance of incorporating long-range dipole component into your scheme, the diabatization scheme?
Excuse me, I didn't.
Well, I'm quoting the question, but basically long-range dipole interactions.
Long-range dipole component. Yes, yes, of course. Like if this is necessary. So actually, maybe the question relates to recent work that has actually been done on nanofibers, where the conjecture is that actually long-range dipole coupling plays an important role. So in principle, one could set up like a generalized scheme where we incorporate this. Now in practice, we might use other schemes. So we actually use different ways of doing a diabatization approach. So I can't promise that this goes like easily and analytically in terms of like this inverse eigenvalue problem, but it's certainly possible. And we're actually now moving to neural network approaches in order to like fit electronic structure information to the desired form of the diabatic Hamiltonian. And the key here is really that we need anharmonic potentials. So it's going to be more complicated than the usual linear Vibronic coupling models. But yes, there's definitely a way to do this.
Okay, maybe Sergei, should we, there's still some questions, but.
Yeah, I see.
Yeah,
yes.
you can actually 5 more minutes. Yeah. About.
Oh, okay.
Yeah.
So I'll ask the question from the chat to everyone. The effect of dielectric constant, how is that considered in the calculations? Yeah,
actually, in these calculations, we looked at isolated chains. So so this is very, very approximate, right? And so essentially, we should include a dielectric environment, which wasn't done properly in these calculations. So so in other types of calculations, we're actually doing this, like especially if we look at exciton dissociation, right? So so we include a dielectric environment in terms of like the effective electron-hole barriers, right? But here we didn't do this yet, but in fact it should be done. And the other thing that should be done is that we get torsional distributions that actually include confinement effects due to neighboring chains. So we believe that this doesn't play a key role, but it hasn't been done properly yet in these calculations. Good question. Yeah.
Okay, I'm going to go to a private question from the private chat, which is, can you comment in a general way on the effects of the anharmonic potentials versus the harmonic potentials of the Frenkel-Holstein? You know, what I guess basically what are the effects of qualitative effects maybe of anharmonicity?
So in this particular case, anharmonicity is actually going to have a huge effect. I mean, these are the real potentials. And so we have a huge effect in terms of the ground state potential of the torsions that affects the sampling and like the Wigner function sampling scheme that we're using. And so actually a harmonic approximation for this potential gives something pretty different. And then on the excited side, harmonic approximation is no good either. So in this particular case, it's actually of key importance to have unharmonic potentials. So there are other cases where like this linear Vibronic coupling model works well, but here it's just far too crude. Now, especially because like in the torsional fluctuations, we can have large excursions, especially in the ground state potential, which is very flat. So unharmonicity is important.
You know, if I can follow up just briefly, so would it be correct to say that, say, the floppier the molecule or the more anharmonic the potential, the faster the exciton diffusion? Could I make that generalization?
Yes, I mean, in a sense, if you look at this picture, right? So the mobility depends on different things. So first, the temperature. Yeah, obviously, we have a huge effect of the temperature. So the torsional fluctuations increase, and we see that the driving of the exciton increases, which actually generates, obviously, the increase in the MSDs and the diffusion coefficients, right? And then the shape of the potentials actually does matter quite a bit. So we did the comparison like between the oligothiophanes and OPVs, yeah, and so the potentials do matter. Yeah. Now the predominant effect we think is actually temperature here, right?
Mm-hmm. Okay. I'm gonna go to David Coker's question. Fluctuations in the interchain pi stacking will need to be described in the electronic structure calculations. How will you incorporate these interchain interactions, I guess, in your future calculations?
Yes, definitely. That's an excellent question. Yes. We have to sample our realizations, right? So we'll start from electronic structure snapshots. Actually, quite a bit of sampling, I think. So we have fluctuations, intra-chain fluctuations, which actually means that the interchain interaction is modulated, and this is definitely what we didn't include here. So this was rigid, right? Mm-hmm. So we have a full set of like intra-oligomer modes here. It's actually a large calculation with 200 modes or so, but so we didn't actually include the modulation of that is due to the interfragment distance, and the effect of the fluctuations of the oligomer fluctuations on the coupling. That's definitely to be included. Yeah.
Okay, maybe we'll stop there, Sergei.
Yeah.
And thanks, Irene, because we really peppered you with a lot of questions, rapid fire.
The next speaker is Frankfurt from University of Rochester.
Can you hear me?
Yes.
Okay, can you see the screen? I have to apologize. After using Zoom for a year, I still don't know how to unmute myself. I would like to begin to thank Suki and Chris for creating this wonderful series and inviting me to give this talk. So today I would like to quickly go through some of the recent adventure we've been making in this new field, so-called Polariton chemistry and molecular cavity quantum electrodynamics. So what I mean by Polariton. So let me give you a very quick introduction in case you never heard about this. So when you couple molecular system into this optical cavity, usually formed by this reflecting mirror, you start to hybridize electronic excitation with photonic excitation and create a set of hybridized excitations, so-called Polaritons. Okay? So these are the usual optical devices formed by reflecting mirror that could be metallic, such that the radiation field within these two mirrors are properly quantized, right? There you can speculate there are only certain amount of frequency of light can be allowed within these two boundary conditions. Now, of course, you can write down a form of Hamiltonian to describe such light-matter interaction. So you can write down a total Hamiltonian. You can write down your Hamiltonian for the matter. You can also write down the Hamiltonian for the radiation field. In this case, I'm only writing down one mode between these two mirrors with a frequency I call omega c, and A and A dagger are the raising lowering operator for the field. You can think about they are photonic creation annihilation operators. And of course, light and matter will interact with each other. In this case, I'm using a dipole gauge Hamiltonian. So the interaction is predominantly carried by the dipole of the molecule and the electric field of this quantized mode. Okay, so that's basically the basic setup. And when you have this light matter interaction, you really have to give up the idea that you should only focus on the molecule or you should only focus on the cavity field, but rather consider this hybrid system as an entity, in the sense that if you want to describe the quantum state of such hybrid system, you really have to extend the size of your Hilbert space. Not only you have to tell me what is the quantum mechanical state of the molecule, but also you have to tell me what is the quantum mechanical state of the photon field. For example, here I'm showing you two potential energy surfaces for this diatomic molecule. The molecule could be in the ground state or excited state. Meanwhile, you should also tell me how many photons do I have in this cavity. In this case, I have 0 photons. Let me quickly do a laser pointer. 0 photons in the cavity. As you can expect, these are the usual potential energy surfaces for a given diatomic molecule. And for the red surface, that means the molecule is in the excited state. And in this community, we call that we created exciton in the molecule. For this hybrid system, you could have different type of excitations like this. So this is a state where the molecule is actually sitting in its ground state. That's why it's adapting exactly the same curvature as the previous one, G0 state. But the cavity is being excited. So there's one photon contained or confined between the two mirrors. So in the sense that this potential energy surface is a surface describing the hybrid system being G1 state. Molecule in the ground state, one photon in the cavity. As you can see, it's an exact replica of the previous curve, but energetically shifted up with exactly the frequency of the photon, right? H bar omega c. So I indicate that as a blue excitation of the cavity mode. You usually don't call that exciton, you call that photon. Now, through the light matter interaction term, Hamiltonian like this, they will start to mix and hybridize and generate a new sets of curves, right? We usually call that the upper and the lower Polariton states. So this is really the Polariton. You can really think about they are the hybridization of molecular exciton and photonic excitation, which is photon. Formally, they are Polariton Hamiltonians eigenspectrum as a function of nuclear position. So of course, this is an interesting field. There are many theorists start to pay attention to this. I'm just giving a list of that, but it's interesting in the sense that the story is, like I said, you're replicating the ground state curvature, lift it up with the frequency of the photon, your photon dressing that state, and start to hybridize with the excited state curvature. So by doing this, you are actually modifying the excited state curvature as well as non-embedicity, such that you can introduce a new minima here, new barrier here, some void crossing, even conical intersections. And photochemical reactivity is all about these different dips and barriers and change of non-embedicities as well, right? So if you use another instant laser, external laser to excite a system, put it on the excited state, excited Polaritonic states, then you'll see that there's a really rich dynamical interplay among all of these degrees of freedom, electronic, nuclear, and photonic degrees of freedoms, such that if the molecular vibrational motion squeeze the system into this region, the system is predominantly sitting in an excitonic state. Right? If it is stretched a little bit, it's predominantly sitting in the photonic excitation state. And the purple is just something in between. You can call that Polariton. So as you can see, just by using the vibrational degrees of freedom, right, the system is constantly exchanging photons and excitons with cavity, right? In the sense that if you stretch it a little bit, the molecule will convert an exciton into a photon. It's very similar to spontaneous emission process, with the difference that here the molecular vibration is modulating these processes rather than the usual thermal fluctuations, right, by the Weizskopf-Bigner mechanism. And this really gives you a chance to significantly change the chemical reactivities using the tools available in quantum optics, because here what you're doing is let them interact, but the dial you can tune is the frequency of the cavity, the coupling strength, or even the quantum mechanical state of the photon. Of course, that sounds like theorist's daydream, right? So I have to convince you that there are a couple of experiments has been done to demonstrate it is possible to change the outcome of chemical reactions. For example, there's this photochemical isomerization reaction can be significantly slowed down, demonstrated by Eberson's group. They also show that by hybridizing molecular vibration with cavity excitation, you can also selectively change the reactivities, favoring one reaction versus the other. You can also create nano objects with these optical devices in the so-called plasmonic cavity. You can even control or create Polaritons at a single molecule level. As a new field, you see this is giving a lot of promises to profoundly change chemical reactivities through the tools of quantum optics. But as a new field, there's lots of ambiguities, and the fundamental limit and basic principles for these mechanisms remains elusive. So what I'm trying to do today is to give you some of our theoretical understanding of these experiments. So I'll first talk about Polariton photochemistry and tell you some of our understanding on how Epperson used the cavity to change the outcome of their photoisomerization reaction. And then we'll talk about vibrational strong coupling, which is also a mystery in Polariton chemistry. All right, let me begin with this. So what Epperson did is coupling this photoisomerization reaction within a cavity. And what he found is if he tuned his cavity frequency into a resonance with one of the reactant, so he can start to generate these Polariton states, he can significantly slow down the chemical reaction in both rate and yield. And our hypothesis, like I said at the beginning, would be because the force exerted on the nuclear degrees of freedom is profoundly changed, right? You're changing the excited state topology. And hence, it's likely that you can change the chemical outcomes just because of the rich dynamical interplay between the electronic and photonic dynamics. And to this stage, we can, of course, jump into a very realistic description of the system, ab initio on-the-fly simulation, quantum dynamics. But we feel it is more beneficial to just begin with simple model system that capture the essential physics and hopefully left out some unnecessary details so we can really understand the fundamental limit and basic principles of this new field. And this is exactly what we did. We built a model system that described isomerization. In the ground state, you have two wells, cis and trans. In excited state, this is a relatively flat potential. This is a usual curve for the S1 state for photoisomerization, because when you photoexcited system, the electron density tends to flatten out and then it becomes more delocalized. All right, so if you do photoexcitation for this isolated system without any cavity, then you should expect roughly 50-50 selectivity. The reason for that is no matter where you start or begin with, you photoexcited system, the wave packs can fully explore the original excited state and it's relatively flat. It doesn't have any preferential directions for the reaction. So of course, after a certain amount of time, when the radiative or the non-radiative decay channels take care of the decay back to the ground state, you roughly get 50-50 selectivity. So you roughly ended up with 1-to-1 ratio for cis versus trans. Now, the story will be changed if you couple the system to a cavity. Remember, the idea was replicate the ground state curvature, move it up with the frequency of the photon, and start to hybridize with the excited state. By doing that, you're creating Polariton states. In this particular case, you're creating a barrier that separates cis versus trans, such that you're stopping the Polaritonic wave packets to visit the other region of the nuclear configurational space. And after the decay channels, you're gonna end up with almost 100% cis configuration. You can also slightly change the cavity frequency such that you generate this set of potential energy surfaces. And then because of the relatively flat original excited state, as well as the new photon dress states and the hybridization of these two, you can really guide the Polariton wave packets to visit the other side, predominantly visiting the other side of the nuclear configuration. So it ended up with almost 100% of trans. And we believe that Epperson's experiments is basically operating in this particular regime where they're stopping the reaction to happen. And we predict that if he changes his frequency a little bit, he will be also able to see that he can facilitate such a reaction. And in fact, you can really start to think about using cavity frequency or the photon frequency as additional tuning knob. So you can have the additional degrees of freedom to change the chemical outcomes. In this case, we're changing from originally 50-50 mixture to almost 100% cis, all the way to 100% trans, just by changing the frequency of the cavity. More interestingly, if you couple more of this molecule with the cavity, then you can start to create collective super excitation of the system, in the sense that in this case, we're using a resonance cavity coupled to both molecules. And as you can see, the lower Polariton eigenspectrum really corresponds to a superposition of molecular excitation and photonic excitation. So my notation here is like this. The first molecule in the ground state, second molecule in the excited state, 0 photon in the cavity, or in this case, ground state, and you have 1 photon in the cavity. And if you do a initial excitation, you can see this is a delocalized excitation among all degrees of freedom. But very quickly, you find that one of the molecules predominantly take the excitation. I summarize, corresponds to a reaction coordinate sliding down on this potential energy surface and emit a photon back to the cavity, as you can see from this yellow curve here. And then a second molecule will take that photon, react, and emit a photon back. So through this process, we actually using just one photoexcitation or exciton and enable 2 chemical reactions. In that sense, your quantum yield for this chemical reaction is 200%. So we actually accomplished something that break so-called the Stark-Einstein limit, which suggests that you have 1 photon per chemical reaction. Here, we're recycling photon as a catalyst because it will regenerate it at the end of the reaction per molecule. And it doesn't have to be a real photon. We even demonstrate that you can do virtual photon exchange between molecules, and that will still enables you to initiate this chain reaction through so-called super exchange mechanism between these excitonic states and photonic excitations. Okay, so we've been having a lot of fun with this Polariton chemistry idea in the past 2 years. And for example, we've been studying this Polariton-mediated charge transfer, where you can expect that by forming Polaritons, plus and minus states, you can significantly change the driving force of this charge transfer reaction, right, from donor to the acceptor, as well as the effective coupling between the donor and acceptor states. Now here, the donor state will be this light matter hybridized Polariton states. And we'll be also interested in Polariton photophysics, and collaborating with our colleagues, Nick von Mickes and Todd Krause. We're trying to understand if you couple lots of these chromophores, in this case, this nanoplates, with a cavity modes, right? How should we properly understand these complex electron, photon, and nuclear degrees of freedom, and the dynamical interplay of those, and why you can only observe a certain photoluminescence at a particular incident angle in this cavity devices. And we've been using theoretical simulations to understand those interesting phenomena. And this field also gave me an opportunity to learn quantum optics and trying to resolve some of the fundamental issues in quantum optics itself. As you can see, I'm beginning with a particular type of Hamiltonian, which is a dipole interact with electric field. It turns out that in quantum optics, there's this gauge ambiguity that's saying, there are actually different type of Hamiltonians, and if you use them, you often get different results. But you should get the same result as they are formally gauge invariant, or they're related through unitary transformation. And this is a problem really bothered physicists for decades, as Janus really said back into the 70s. He said, how could you ever trust your result if 2 gauges or 2 Hamiltonians generate different results? But beyond that, how could you leave with this shadow that for a gauge invariant theory, formally, it breaks down once you apply them to 2-level atoms. So we really have some chance to sit down and think about this during the lockdown last year. And what we find is it's because not all terms in a Coulomb gauge Hamiltonian are properly truncated or constrained within the electronic subspace. And the resolution for that is you can reconstruct a proper gauge transformation operator to do that job. And at the end, you have gauge invariant Hamiltonian at any level of electronic state truncation. So as you can see, this is a little bit more complicated because it doesn't correspond to a simple modification of Coulomb gauge Hamiltonian, the minimum coupling Hamiltonian, by changing its momentum matrix element, but rather profoundly change the light-matter interaction structure in this Hamiltonian. But we're glad that it gives us a chance to finally solve this long-lasting ambiguity. So based on what I said, I think this is an interesting field because it allows you to use quantum optics tools to tune chemical reaction, so you can change the reactivities. And it works the other way as well. It gives you the molecular degrees of freedom, so you can really think about how to take advantage of molecule and do interesting photophysics or quantum optics. For example, in quantum optics, there's really this interesting phenomenon, so-called dynamical Casimir effect. What it does is, what it predicts is basically if you rapidly moving your mirror or vibrating your mirror, because the Fock state before and after you moving the mirrors are no longer orthogonal, and due to this non-orthogonality, you can really generate multiple photon pairs just from thin air, or by fluctuating the radiation mode, the vacuum radiation mode between the two mirrors. This is a really interesting phenomena. Experimentally, if you want to realize this, you have to move your mirror so fast, approaching to the speed of light. But with molecular system, you can really think about to achieve the same goal from a different perspective. For example, you can really think about take advantage of the molecular vibration as that degrees of freedom instead of fluctuating the mirrors, using with speed of light. And what you need is two electronic states that have quite different permanent dipoles. And by that, if you look at the Fock state associated with each one of these electronic states, they are largely displaced, and hence they're really non-orthogonal. And through this non-orthogonality and the additional nuclear degrees of freedom, you can achieve the same goal as what has been originally proposed in this dynamical Casimir effect. So what we have here is basically for the covalent ionic states in a typical diatomic molecule, we can let them couple to each other when they're having different Fock states, and the Fock state associated with these two states are not orthogonal to each other. And of course, you can definitely generate multiple photon pairs just by starting from one initial single excitation. So basically, you excite the molecule and let the vibrational motion as well as light matter coupling take care of that. You can generate multiple photon pairs. So like I said, this is really giving the quantum optics community also a brand new tools, and start to think about how should you take advantage of a molecular system and a molecular structure and enable new phenomena. All right, with the rest of my time, I would love to quickly touch base upon this vibrational strong coupling and Polariton chemistry. So this is a very interesting set of experiments where you don't couple electronic excitations with cavity, but rather you're coupling you're coupling vibrational excitations with cavity. So for example, you have a ground state, you have different vibrational levels, and then your photon frequency in the cavity is matching those vibrational excitations in a sense that you can have one photon dressed vibrational levels and bring that into a resonance with the original excited vibrational state. And they start to generate hybridization states and split and gives you the polariton. So these are so-called vibrational polaritons. And you can really measure those vibrational polaritons through IR spectrum or transmission spectrum like I'm showing you here. Now, the fundamental question is, would this interesting generation of vibrational polariton also translate into or manifest itself into the change of reactivities, just like what we see in the polariton chemistry, photochemistry cases? So it turns out you could do that. And I have to remind you that when they do this chemical reaction, there is no external laser excitation whatsoever. Everything is governed by the thermal fluctuation of the vacuum field. What they find is indeed, if you have that cavity and if you tune the cavity frequency into a particular one, into resonance with the system, you can selectively slow down the chemical reaction rate by almost 4 times. And this turns out to be a mystery in the theoretical chemistry. We don't have a good theory to understand what was going on. So my intention back in the last summer is really trying to build a model system, again, simple model system, and hopefully I can understand a little bit of fundamental physics out of it. So what we did was trying to couple this Shin-May-2 type of model, which is transferring both electron and proton between the 2 fixed nuclei, and then couple that with the cavity. I'm using the Hamiltonian I'm writing down at the beginning as well. In this case, I'm only having the ground electronic states, but that permanent dipole associated with the ground state is coupled to the cavity. So the total Hamiltonian looks like this. And I can also visualize a so-called cavity Born polymer surface. It looks like this. So here I'm printing out both the electronic component as well as the photonic component. I'm leaving out the photonic momentum part of the Hamiltonian as well as the nuclear momentum of the Hamiltonian. So I can visualize this potential along both the reaction coordinate as well as the so-called photonic coordinate. With that, it turns out that in such a simple model system, we don't really understand what's going on. Like I said, as these interesting papers, they all pointing to one simple conclusion. There is no modification whatsoever from the cavity to those vibrational strong coupling cases manifesting to the change of reactivities. No effect at all. Or, you don't see resonance effect. Basically, you can change your cavity frequency and there's no significant change of the rate constant, right? In contrast to the experiments where you can see a slowdown of reaction rate by 4 or 5 times. So my intention is, can I resolve these mystery from simple calculations? And here we go. So basically, we have this Hamiltonian again. I'm showing you a normalized coupling strength, which is the Rabi splitting divided by twice of the cavity frequency. And that really indicates how strongly light and matter interact. So here I'm showing you again the Polariton potential energy surface. And I can change the coupling strength from weak to strong. In this case, actually very strong. As you can see, what this Hamiltonian tells you is, no matter how strongly you couple this molecule with the cavity, it does not change the relative barrier height from the minimum to the top of the barrier. It just doesn't change. The reason for that is, in this light matter interaction, you always have a quadratic term, a complete square. So as you know, it doesn't change the absolute value of energy, but rather it only displays the minimum along this photonic coordinate. That's what you see here. Notice that the scale of the coordinate is different than this one. So that's why if you just do simple transition state theory, it doesn't give you any effect, and it shouldn't, because that's what the light matter interaction Hamiltonian tells you. And hence explain why in the previous cases, when you're just using transition state theory, you didn't see anything. Of course, TST is only a crude approximation to the rate constant. If you want to have a full description, you should also worry about dynamics, right? Remember, in Gen Chem, when we learn transition state theory, the fundamental underlying assumption is once you reach to the transition state dividing surface, the reaction will go one side. There is no recrossing. So what is missing is actually the recrossing, and it is captured by this so-called transmission coefficient. And you can formally calculate this through this flux side correlation function, where you just choose a dividing surface and a side function, and the flux is the time derivative of that. So we can compute this either quantum mechanically or classically. And here we have
Frank, sorry to interrupt, but your time is already up. Yeah.
Okay.
Wrap up in 1 minute. Yeah.
Yes, let me wrap up. So what you can see here is basically transmission coefficients is the cost of this slowdown, selective slowdown of chemical reaction. Where you see, as you increase the coupling strengths, the plateau value of these transmission coefficients keep decreasing, and they become more oscillatory. And you can also print out reactive trajectories from these ensembles, and you will see that when you have really large light matter interaction, you are actually forming a solvent cage such that the reactive trajectory is selectively trapped or slowed down in this region. There are more recrossing. You're facilitating recrossing. It turns out this is really a classical picture of solvent caging effect that has been discovered when people study SN1 organic reaction in a solvent, why solvent could selectively slow down a given chemical reaction. In a textbook example, I really rip up this page from a textbook, and you can see a reminiscent of those pictures as what I'm showing you here. So our hypothesis is cavity mode is playing the same role as a regular solvent, such that when you couple that with reactants, it formed a solvent cage at a certain frequency, and it can selectively slow down your chemical reaction. And of course, just with such simple model system, we can almost recover, the essential features discovered in the experiment. So this is really an interesting hypothesis. Let me quickly summarize what I've been talking about to you today. So for the Polariton photochemistry, I demonstrate that you can use the tools of quantum optics to control the chemical reaction outcome, and vice versa. You can use the molecular structure to really think about changing the field of quantum optics as well. And in this vibrational strong coupling Polariton chemistry, I try to explain what is the origin of the reaction constant reduction effect in those experiments, and we conclude that it's predominantly coming from the dynamics, the recrossing dynamics, and how cavity modes can forming a cage, solvent cage, effective solvent cage, such that it will selectively slow down the rate. With that, I would love to I'll quick skip that, and would like to thank my group who did the work, especially grad student Arkady Mandel, Xinyang Li, and Mike Taylor, who contributed to the work I talked about today, founding resources, and thank you so much for listening. I would love to answer any questions.
All right, thanks, Frank, for a great talk. Sergei, what's our timeline?
2 minutes.
2 minutes?
Yes.
All right, Frank. So what about conical intersections? So you modify the excited state potential energy surface, but what is the effect on the the conical intersection coupling back to the ground state? That's the first question. I'm going to rapid fire.
So yeah, so conical intersection. So it turns out you can either create a new conical intersection, or you can split one conical intersection into two. So there's a recent work by Sean Buchmel. If you split that into two, the photodynamics is more complicated. If you generate a new conical intersection, then you can, of course, rapidly facilitate the population transfer from excited state to the ground state. This is really the interesting thing about Polariton chemistry, because you now have additional degrees of freedom, where is the molecular orientation with respect to the field polarization direction, and that angle is a new different degrees of freedom. So in a sense that you can even create a conical intersection, you can engineer a conical intersection in a diatomic molecule, which in principle you should not have.
Okay, and then one last quick question. A lot of these cavity experiments are done in thin films rather than isolated molecules. Do you worry about changes in potential curvatures just due to the fact you have this concentrated thin film instead of an isolated molecule in space?
Yeah, that's a very good question. So all the stuff I'm showing you today is based upon single molecule coupled to the cavity, or few molecules coupled to the cavity. When you have a thin film, you really have a lot of molecules collectively coupled to the cavity field. And the picture there is slightly different in the sense that you're coupling the collective bright excitations of all these molecules in the thin film with the cavity mode. So you have upper and lower Polariton states, but you also have a bunch of dark state in between in the idealized picture. Now, if you consider also the phonon fluctuations, you actually have something close to that picture, but not exactly to that picture. And that actually profoundly changed the way you can measure these spectroscopy responses, like the fluorescence. You see that the emitter will predominantly fluoresce at one branch of Polariton states, not the other. And then you can only observe certain fluorescent signals at particular incident angles. And we've been looking into this. For the chemical reactions, this is also a good question in the sense that chemical reaction often happen locally. Whereas when you create those excitations, it's pretty much delocalized among all the molecules and the cavity, right? So it is still a mystery that how a localized chemical reaction could be profoundly influenced by the delocalized Polartonic states, created by the light matter interaction. So I have not. We're working on it. We haven't got a chance to fully resolve it, but I think it itself is also a very exciting field.
Okay, maybe we'll stop right there. I encourage actually both Frank and Irene, there are actually still questions outstanding in the chat, but we're going to have to move on to our next speaker, experimental results probably from Gabriela Schlau-Cohen from MIT. So Gabriela, take it away.
Great. Thank you, Sergei and Chris, for the invitation to come and share some of our work with everybody today. So everything that I'm going to talk about today is in close collaboration with two of my colleagues here at MIT, Mark Botte in the Department of Bioengineering, and Adam Willard, also in chemistry. So there will be some theory in my talk as well. So a lot of what brings us together today is that excitons underlie a huge number of processes: biological light harvesting, solar energy, display technologies. But controlling and optimizing these excitons, or even understanding them, is challenging in condensed phase, because if we zoom in, we can see just how crowded these systems are. And so this crowding leads to a range of different interactions. And as these systems evolve in time, they move on a range of different timescales. So all of those degrees of freedom lead to a really difficult problem. So what we want to think about is, can we construct a material that allows us to control these excitonic systems? And so that's the overarching goal of the work that I'm going to tell you about today. And the approach that we take is to use DNA, where we construct an excitonic system, like the one shown here, where there's a DNA scaffold, and we covalently attach different pigments, different dyes within that scaffold. And so we're not the only ones to do DNA excitonics. There are several different groups. Some of them are shown here within this space. And the reason that so many people are excited about this area is fundamentally because of the flexibility of DNA. So we use a technology called DNA origami. And for those of you who aren't familiar with it, I'm going to illustrate it using a movie. So how DNA origami works is you exploit the highly specific base pairing of DNA. A binds to T, C binds to G, and you design your strands so that when they anneal with complementary strands, they form an arbitrary nanoscale 3D structure, such as the tile that you see emerging before your eyes. So what we can do is we can exploit that ability to control nanoscale architecture and position within that nanoscale architecture at desired sites dyes. And so what we want to think about in using this platform and zooming in, so we want to think about, can we control this system, or our total Hamiltonian? We can think about this in the typical two halves: the system parameters, or our electronically excited dyes, and the surrounding environment, often referred to, of course, as the bath. And like everyone, we tend not to care about the bath itself, but rather its influence on our system. And so if we're thinking about our system, we can think about the electronic transition energies, which are controlled by the molecular structure, the choice of the dye, and then the intermolecular couplings between the dyes. And when we think about the interactions within the environment, we can, somewhat arbitrarily divide it into two pieces: the local effect due to rapid fluctuations, the short time dynamics of the environment, and then the influence of slow anharmonic motions that can shift the energetics of our system, or the long time dynamics. So here's one version of the Hamiltonian that we can think about. We've already seen versions of this approach today. And so what we want to think about is, can we begin to tune these individual pieces? And it is particularly challenging to think about disentangling the effects of the environment because of the many degrees of freedom that I showed you in the bath. So if we want to think about constructing a controllable system, we want to think about how we can tune those knobs, and that challenge of how we can understand and use the environment to influence the function of the excitonic system. So these are the specific questions that we addressed in the work that I'm going to tell you about. So first, thinking about tuning these knobs. So if we take our DNA and we covalently attach within that dyes, in this case, Psi3 or a modified Psi3, we can build a dimer system. We can characterize the electronic coupling of the dimer using 2D electronic spectroscopy. So this is an ultra-fast technique. I'm not going to go into how we do it, but basically what you get out is a correlation plot where you correlate excitation energy and emission energy. And where you see a peak that correlates excitation and emission energy, that is a signature of electronic coupling between those two excited states. So we can use 2D spectroscopy to measure the electronic coupling. And we take this approach within our systems, and we take these DNA constructs and we increase the distance and change the relative orientation within our dimers. We repeat this measurement and we see that signature of electronic coupling decrease. We can pull out the intensity of that feature, and the relative intensity is shown in gray, and we see that decrease in electronic coupling. We can compare that to calculations. These are the results of transition density cube calculations on MD structures that were done by Will Burghardt at the University of New Mexico. And we see that we have general agreement between our experiment and our theory. So that tells us, in terms of tuning these parameters, we can control the molecular structure, the electronic transition energy, through the dye we choose to covalently attach. And we can control the electronic coupling systematically within dimer structures. So now thinking about the influence of the environment, we can actually go back to our 2D spectra, because these are not just correlation plots at a single time, but we can measure the correlation as a function of the time delay between excitation and emission. And so at short times where excitation and emission are the same, we see a near diagonal line in our peak shape. And then as those energies fluctuate, that peak becomes rounder. And we can quantify that using the slope of that feature. And so that slope is proportional to the correlation between excitation and emission energy. And here are representative time points that show that evolution. And so what this is an experimental measure of the frequency-frequency correlation function. So that data is shown in this plot here for a monomeric dye on a DNA duplex, and then for a free dye in solution. And we can analyze this frequency-frequency correlation function and pull out two things. We pull out the exponential decay timescale, the overall timescale of spectral diffusion, and we see that becomes faster within the DNA. And we also see oscillations within this, which we can Fourier transform. We see a peak at around 450 wave numbers. So that tells us about a 450 wave number oscillation within the dye that is an underdamped mode coupled to our local transition. So overall, what that analysis told us is that in terms of the short time dynamics of the influence of the environment or the bath, we see that that time scale increases within that DNA scaffold. Now, if we want to think about the longer timescale dynamics, what we can use is single molecule spectroscopy, because this allows us to measure the distribution or the inhomogeneity within our system due to the long timescale or anharmonic motions of the environment. And the measurement we use is the fluorescence lifetime, because this is a way to capture the overall photophysical pathways within an excitonic system. So to begin with, we take a monomer, 1 Psi3, on a DNA scaffold. We measure the fluorescence lifetime for one, then another, and then another, and we build up the distribution of lifetimes. And what we see here is that for a single Psi3 dye on our DNA scaffold, the distribution has a width of nearly 1 nanosecond. What we can do is we can change the scaffold. So we switch it so that that dye is, instead of just on a duplex, on what's known as a DX tile, illustrated in this cartoon here. And that DX tile has a persistence length that's twice that of the duplex. And what we see from that change is that the width of this distribution narrows to 0.2 nanoseconds. So this data is for a monomer on the DNA. We see the same trend for a dimer on the DNA. And so this tells us that we can use the scaffold to control the width of the distribution of photophysical properties, or which is related to the inhomogeneity of our system. And we know that those differences in lifetime are coming to differences in the exposure of the Psi3 to the solution, because it's known that Psi3 lifetime is dependent upon the exposure to the solution. We can also measure the time scale of different fluctuations using fluorescence anisotropy. So these are the anisotropy fluorescence decay curves for a monomer on a duplex and then on a DX tile. And by fitting this within a simple cone angle, what we see is that actually this tells us that those structural fluctuations have different magnitudes within the time scale of the lifetime, where the DX tile has almost 10 degrees larger of a cone angle. So it tends to, although the width of the distribution is narrower, tends to sample a larger cone angle of fluctuations. And so this tells us that through the DNA environment, we can control the fluctuations within the system. In this case, we're seeing that we can control the magnitude of the structural fluctuations. So in thinking about tuning these parameters, what we see is that we can influence the photophysical heterogeneity, or essentially the long time dynamics and their influence of the bath, as read out by the fluorescence lifetime, as well as the magnitude of the structural fluctuations. And we can do that through selecting the properties of our surrounding DNA scaffold. Now that we begin able to explore to what extent or how we can begin to tune these knobs, we wanted to think about, can we use that at all? Can we think about how the environment might be able to influence the function of our excitonic system? And so what we were particularly interested in is thinking about exciton transport. And so we looked at that by looking at energy transfer, where we took our Psi3 covalently attached within DNA, and we added a trap, an acceptor dye, a Psi5 acceptor. And then we looked at the efficiency of energy transfer from Psi3 to Psi5. And what we saw is on the duplex, we saw a very high efficiency, around 95%, from a monomer Psi3 to the Psi5. We then repeated that on the DX tile, and we showed that the energy transfer efficiency dropped by around 20%. So this result was really surprising to us. We confirmed it through several different types of measurements, steady state fluorescence, time resolved fluorescence, because we have two dyes which are chemically identical. The donor and the acceptor are chemically identical in both of these systems. The average distance that you would predict from a straightforward calculation is the same. So the distance is the same, the dyes are the same. So we were surprised to see such a dramatic change in the efficiency of energy transfer. And we repeated this within the dimer, and we saw roughly the same effect. The absolute magnitudes are a little different because it's a H aggregate-like, so the quantum yield decreases a little bit. But we saw that again, in the same case, these dyes that are chemically identical actually have different, and the distances are the same again, have different efficiencies of energy transfer. So to think about what might be going on, we can return to this fluorescence anisotropy. And what we see is within this anisotropy decay, that not only is the magnitude of the cone angle different, but the time scale of the anisotropy is different, where for the DX tile, the structural fluctuations are slower. There's a 0.62 nanosecond timescale of the structural fluctuations. We can contrast that with a duplex. It's more flexible, it has a shorter persistence length, and consistently, the timescale of those fluctuations are faster. So if we think about the timescale of energy transfer, that tells us that the DX tile is roughly in the static limit, where these structural fluctuations are on a longer time scale than energy transfer. And the duplex is in roughly the dynamic limit, where those structural fluctuations are faster than the energy transfer. So why does that matter? Within a simple model, where you just take sort of a centered, a cone angle model, then whether you're in the static limit or the dynamic limit, your efficiencies are roughly the same. But if we return to our MD trajectories and look at the actual distributions of the angle of our donor, what we see is actually the distributions are structured. This isn't a simple system with a Gaussian distribution centered around a single value. We have a structured distribution, and actually particularly in the case of the DX tile, as you can see here. So there are subpopulations. So when you're in the static limit, so in this, you have those subpopulations that you maintain throughout your energy transfer time. And so we see that this condensed phase environment gives rise to this structured angular distribution. And you essentially get stuck in those subpopulations within your DX tile. So when you have these slower fluctuations, and then this is the theory, we can take the theory we can simulate it using that distribution. In the dynamic limit, we're able to replicate that high efficiency. And then in the DX tile, in the static limit, due to the presence of those subpopulations, we lower that overall efficiency. So where this difference in efficiency is coming from, even though they're chemically identical, is due not just to differences, is due to differences in the angle, and not the average angle, but actually the distribution of angles and the time scale over which you sample those angular distributions. And so what this is showing us is that within the static limit, you can have some subpopulations that lower your overall efficiency. And why we thought this result was pretty surprising is often when you think about building a system that, that transfers energy well and reliably, you think about wanting to suppress fluctuations. You think about fluctuations maybe, can enhance energy transfer, but overall you want a system that's well defined. But actually it's the more fluctuating, rapid, flexible duplex system that is able to transfer energy more efficiently than the DX tile. Because in the DX tile, those subpopulations end up lowering your overall behavior. And so in thinking about this question, tuning these parameters, we were able to see that the long timescale photophysical and structural heterogeneity was present. And not just that there were differences in the structural heterogeneity, but there were differences in the timescale of the fluctuations that associated with that structural heterogeneity. And that difference in time scale was actually what was able to cause a 20% difference in the efficiency of energy transport. And that the case of rapid fluctuations actually enhanced that efficiency of energy transfer. And so hopefully what I've illustrated with this representative example here is that by using DNA origami, we can precisely control not just the position of dyes to control their electronic coupling and interactions, but they actually control the properties of the surrounding environment, such as the scaffold, varying those interactions to influence the dynamics of an excitonic system. Okay, so I thought I had too much, but apparently I went too fast. So I'm done, but I do want to glad I have time to thank the people who actually did all the work. So all of the spectroscopic experiments that I showed you were done by Steph Hart and Weijia Chen, two great graduate students in the group. The design and production of the DNA origami systems was done in the lab of my colleague Mark Bate, as I mentioned, and James Banal, a postdoc in his group, who's kind of delocalized, in fact, between our groups, has done a lot of the work developing those systems. The simulations were done by Adam Willard, along with his graduate student, Amr Doden, and then Will Bricker did a lot of the MD structures and the calculations of the coupling. These are the awards that keep the lights on, and I am happy to answer questions.
Great. And I compliment you. We have time. We do have time for questions. And so we will just jump into them right away. So the first question is the faster decay of the frequency correlation function in the DNA duplex seems to be faster than that in solution? Is that counterintuitive?
No, because you're measuring really the influence of the local environment. So if you think about the local environment when you're in DNA, you're covalently attached to this charged system. So because you have all of the charges in the DNA and the local counter ions, those really nearby ions, any kind of motion or fluctuation in them is likely to have a dramatic influence. So you expect the collective action of them to lead to a faster decay. So in retrospect, it actually made a lot of sense, where it's the longer time scale behavior that may be hindered, or fluctuations that may be hindered by the environment, but the short time scale is actually going to be faster.
Okay, the next question is, I think this is referring to the DNA duplex versus the DNA tile. What about the local dielectric? Could that play? I think the question is basically, could that play a role in the different energy transfer rates?
Yeah, we expect that to be the same, because we actually did this experiment with the exact same sequences between the Psi3 and the Psi5. So we designed our strand so that the sequence was the same.
Okay.
So it actually took a couple years to kind of nail down exactly what was going on. We had to try many things. Okay.
Yeah, you know, there's always this sort of debate. What is the n that you put in the n to the fourth? You know, sort of. I think it's n to the fourth, right, in the Forster transfer expression. So if you think that your n is different, I guess it could have a big effect. But nobody really seems to know.
Yeah, absolutely. And you know, when you have all of the charges, like I was talking about with DNA, that becomes hard. But that's why we went to so much effort to make sure that that what was between these two dyes was the same, so we could think about these things comparatively.
Okay. So another question: what is the time scale of the angular fluctuations? You know, do you have any handle on how quickly those are changing?
Yeah, so that is what was measured with the anisotropy, the angular fluctuations, which was in the DX tile 0.62 nanoseconds and in the duplex 0.12 nanoseconds. So it's somewhat comparable to the energy transfer rate, but essentially on other sides, so that we can kind of conceive of them in the static and dynamic limit.
Okay, and then another question, in the energy transfer, did you look at the fluctuation? You know, we're talking about the angular deviations, but what about the fluctuation in the donor-acceptor separation?
Yeah, so I think we looked at differences in the donor and acceptor fluctuation between those two constructs, and we looked at fluctuations, but they seem to be very small, too small to accept that. I mean, we didn't look at it experimentally. We analyzed the MD trajectories to pull out some differences in the fluctuations, and they were, you know, an angstrom-ish, so there seemed to be very little fluctuations. And then for the acceptor, you have a little bit more fluctuations, but that's sort of, again, similar and well described by essentially a cone angle, because it's on a floppy linker.
Okay, so the DNA is doing its job, I guess. Another question, again, about the simulations in those, or in the measurements, in the SMS measurements on these systems, do you ever see configurations that could give you coherent electronic energy transfer? Is there any evidence that, you know, there's some magic configurations, I guess?
So I think coherent energy transfer wouldn't happen between the donor and the acceptor because the distances are too long there. One could conceive of coherent energy transfer within that dimer. It's relatively strongly coupled. You could conceive of coherence or coherent energy transfer within that system. So we looked for oscillations. That was kind of one of the goals of this work under the call that we were funded by, and we couldn't find any. So our hypothesis is that the charge of the DNA is actually destroying that phase coherence, because same way it leads to a rapid decay of the frequency-frequency correlation function, the charge along the backbone of the dye is likely to be influenced by that. So we've started to think about some other dyes that may exhibit more robust effects in terms of maintaining phase coherence between our states.
Okay. So with that, I think I actually have more questions, Gabriela, but I think we'll move on to our next and last speaker, which is Sergei. If you want to share your screen.
Okay, great. Thank you so much. And let me start with thanking Sergei and Chris for organizing this fantastic seminar series and for letting me speak here. And without any further introduction, I will jump into my talk, where I will continue. The theme of today's session is how can we understand the coherent electron vibrational dynamics in organic chromophores. So again, we are talking, this is a pictorial picture showing you we are exciting. Then the initial photoexcitation wave packet goes through the evolution of multiple excited states. And on the way, you could see a bunch of conical intersections, different curvature of potential energy surfaces, and avoided crossings. And there are, of course, coherences, which would be a major topic of my talk. And at the end of the day, we would like to make up something useful. We would like to make the light emission, or separate the charges to reach higher photovoltaic efficiency, or to dissociate the molecule to control the chemical reactions, as we have heard many times today. So during this dynamics, there is an active exchange between electronic system and vibrational system in terms of energy. So in fact, electrons are hitting the system gradually once you go through the non-radiative decay. And during this time, there are a bunch of processes, such as, for example, here you'll see the energy transfer process coming between the end of BDP systems. And this is a case when the simple Forster energy transfer is inadequate. And here the coupling between donor and acceptor is modulated by fluctuating transition dipoles. That's why sort of like it's kind of connecting to the previous questions in the session. And you could represent this electron vibrational dynamics in many cases. As Irene has been talking about the two-dimensional representation of the transition density matrix, this would be our electronic degrees of freedom. That would be our vibrational degrees of freedom. And there is a concerted dynamics between electronic and vibrational degrees of freedom. And if you'd like to talk about coherences, then we could sort of like in hand waving argument to link it into the breathing of the wave function, which goes through the localized and delocalized states. So this is a brief outline of my talk. First of all, I will introduce you into the non-aerobatic excited state dynamics modeling. This is our main tool, how we trying to take into account essentially all electronic and vibrational degrees of freedom subject to a certain approximations. And then for practitioners, I will be very briefly outlining the difference between when you need to use Ehrenfest surface hopping and more accurate multi-configurational Ehrenfest with ab initio multiple cloning. And I'll go to the application, and I will show you the coherent passage through the intermolecular conical intersection, a result that has been obtained in collaboration with our experimental group in Oldenburg in Germany. And finally, I will go into another application on how can we use free electron laser spectroscopy, or true Kars-Raman, for monitoring the coherent electronic dynamics. And of course, I will not have any time for machine learning. So as we have heard today many times, that we are talking about non-aerobatic transitions between excited states, and modeling this is very complicated and computationally expensive. So in our group for a number of years, we have been developing machinery and the code, NextMD code, which allows to address the 1 billion time increase in computational complexity when you're trying to model the non-aerobatic dynamics. What is important that it has been developed to include various commonly used non-ergodic dynamic simulation algorithms such as surface hopping, Ehrenfest, and ab initio multiple cloning. And the trade-offs that we are putting in those simulations is that we are not trying to come up with very sophisticated electronic structure description such as CI or a couple cluster or even density functional theory, but instead we are using simpler semi-empirical Hamiltonians, which are still giving us the opportunity to give a qualitative view on the electronic states. And again, I mentioned the machine learning, but now we can really feed the semi-empirical Hamiltonian models to give the quantitative answers for description of the electronic structure if we'll do the tuning very properly. One of the things, there was a question before, what about solvent effects? So there are two aspects into the solvent effects. Number one is that one could use the standard things like a polarizable continuum model or envelope it into the QMMM, quantum mechanics, molecular mechanics, explicit solvent. And this is working and gives, honestly, a minor effect on the results for the non-ergodic dynamics. However, once you start talking about transition between different states, such as charge transfer to excitonic transition or vice versa, then it is extremely important to invoke so-called charge-specific solvation. When you solvate your system, which is concomitant to the dipole of the states where you are currently on, and this is a subject of a completely different discussion. So again, just to introduce into non-ergodic dynamic simulations, the most common approach would be surface hopping approach due to mostly to Lattice contributions. And essentially, there are many flavors of this, but this is a trajectory basic approach, which I personally sort of like qualify as an ad hoc approach. And at the end of the day, you are not able to model the coherences. In contrast to this, something more complicated approaches such as multi-configurational Ehrenfest approach, when you are running an ensemble of the Ehrenfest trajectories and you are attaching the Gaussian wave packet representing the vibronic wave function to every trajectory, so that at the end of the day, you are able to recover the entire wave function of electronic and vibrational system. And all those coefficients is just showing you the expansion in terms of the number of trajectories and the excited state, adiabatic excited states. This approach is due to Dmitry Shalashilin, our active collaborators. And I'll say upfront that this approach is about 10 times more expensive compared to the typical surface hopping or typical Ehrenfest simulations. One problem with the multi-configurational Ehrenfest goes back to the Ehrenfest. So this is a mean field. And frequently those trajectories that are sampling in the mean field, which is going in between of the two potential energy surfaces, are sort of like sampling the wrong part of the phase space. It's unphysical phase space. So instead, to improve this, again, the idea goes to Todd Martinez and to Dmitry Shalashilin. We could introduce multiple cloning. What it means is that once our Ehrenfest trajectory hits the crossing, such as avoided crossing or conical intersection, we are looking if the curvatures of those surfaces are sufficiently different so that our original wave packet is splitting into two. So we have a cloning effect, and then sort of like we could continue the evolution on two different potential energy surfaces. And the cloning allows us to sample much more efficiently the phase space for the evolution of electronic and vibrational wave function. So lately we have been entertaining a lot this approach. So now it is important that how can we compare those approaches? Put together surface hopping versus Ehrenfest versus multi-configurational Ehrenfest. And we have been recently done some massaging and direct comparison. And just sort of like without going into too much of the details, I'll just give you very rough conclusions. So number one, if you are looking to the population evolution and non-radiative relaxation in terms of populations, look no further than the surface hopping. Of course, this is a very much affordable methodology. And sort of like you need just to make sure that you reach the convergence in terms of the trajectories and you sample properly the conformational space. Of course, there is this problem with the decoherence corrections in surface hopping, and you could introduce those decoherence corrections in various flavors. However, the effect of those improvements is relatively minor compared to the choice of the electronic structure methodologies. Your decay rates would be much more sensitive to electronic structure methods used due to the modulation of the gaps. Change of the gap between excited states by 1/10 of electron volt can change your decay rate by the order of magnitude. That's for sure. So, however, if you would like to look at the coherences as we have been discussing in this session, then both Ehrenfest and surface hopping are inadequate methods. And I will not go into the theory behind that. Instead, you need to use something like a controllable approximation, such as ab initio multiple cloning. That's it. And with that, let's sort of like to jump directly into description and various applications. And my first application would be about coherent electron vibrational dynamics. And as I have alluded to you before, that this vibration include periodic expansion and contraction of the wave function. And later, I will give you a little bit more rigorous definition of the coherence. It goes back to Irene's definition in terms of the density matrix of diagonal elements of the density matrix. So, and you could see in this movie, you see the dynamics of the dimer, and you have a wave function that is coming up and down, delocalizes until sort of like the excitation will self-trap itself. And this is just a hand-waving argument, what typically you will get in the description of such a coherent dynamics. And again, sort of like we have been talking a lot about using this coherent dynamics in terms of helping and directing the energy transfer events. And this goes back to the discussions that we have been having in this session. So my first sort of like application would be to the system, which is a small molecule, donor-acceptor, donor-acceptor structure. And these small molecules have been entertained in the group of Gibazan. And at some point, they have been reaching the world record in the photovoltaic efficiency for OPV devices, around 8% back 2003, 2005. However, those molecules are a well-defined system to probe the coherent dynamics. And that's where our collaboration with Oldenburg is coming. And Antonietta Di Cio is now in the audience, and I will direct all the questions to experiment to her on how to do the two-dimensional electronic spectroscopy. So essentially, this is a molecular structure. We are looking at the thin film. And remember, in the thin film, we have mostly sort of like packing in both H and J aggregate formation. And the bottom, the low energy peak is essentially would be the corresponding to the band gap transition in those systems. Again, goes back to Irene stock. However, sort of like there are many states. And the many states due to interactive individual molecules. And essentially what we would like to do in experiment, we would like to photoexcite the wave packet on the higher potential energy surfaces within this lowest peak, and to monitor how this wave packet is relaxing and what kind of coherent motion will be initiated in the system. Again, this is a slide that is describing the two-dimensional spectroscopy analysis that has been beautifully done in Oldenburg. And once we will monitor the evolution of the off-diagonal peaks, then we could track down the evolution of essentially two main excited states. So just look at this picture, and there are two states, and those states are coming into the crossing within like 40 femtoseconds. And then sort of like we have all the signatures in the 2D spectra that we are going into the conical intersections. And all those wiggles in the two-dimensional spectra point us that there are coherent vibrational dynamics, as we have been discussing in this section. So now, if you would like to model this, you need to understand what are those states. And if I will look at the single molecule, then there is only a single band gap states, and the above states are essentially dark. So within the experimental parameters, you will have an evolution of essentially a single excited state without any possible existence of the conical intersections. And our hypothesis was that, hey, we have a solid state, we have packing of the molecules, and we consider a common case of the H aggregates when you have a dimer, and the upper state of the dimer, which is a coherent superposition of the monomeric states, is optically allowed. And this would be our states from where we are starting our dynamics. Again, sort of like a model system, what can be possibly observed in the solid state of coupled molecular chromophores. And I'll tell you upfront that if you look at the solution of the molecules, then the two-dimensional spectra would be very different, and there are no such signatures of coherent dynamics and conical intersections, which gives us some firmity that this sort of like a model is perfectly fine. So that at the end of the day, once we are looking at the dynamics of this, starting from the dynamics of the coherent superposition, H aggregate dynamics, then we, as expected, will see the slashing of the wave function, and we see the passage of the conical intersection within like 100 femtoseconds. It depends on the number of trajectory, and this is a superposition across all of the trajectories. Now, what is important is that once we start talking about this conical intersection is very different from what we have been seeing before, for example, in retinal, when we are flipping the tail and we are crossing between two states. Here, we are talking about so-called intermolecular conical intersection. When we have two molecules which are not connected by any of the chemical bonds. However, these molecules are interacting electrostatically. They have strong electronic interactions, and the strong electronic interactions is making the potential energy of those two states so that these potential energies have all the signatures of multidimensional states which cross and form the conical intersection seam. So that compared to the intramolecular conical intersection, which are sort of governs the photochemical reaction, we believe that intermolecular conical intersection would be a very common thing in the solid states, in the ensemble of molecules. And the coherent dynamics that is happening through those intermolecular conical intersection would essentially sort of like direct the energy transfer and could be served as a tool to control the energy transfer, its sort of like direction and time scales. So this is essentially the conclusion that is coming from this study. Let me go to the next subject, to the X-ray spectroscopy. And X-ray spectroscopy has a lot of promises. That's the latest tool, for example, that to me, by looking at the simple theoretical view, that I could say that this is a tool that allows us to resolve the dynamics both in space and time. For example, recent Nature Materials paper have been resolving the Polariton dynamics in hybrid perovskites. So, and also the X-ray spectroscopy has a lot of promises to resolve the coherent dynamics. But how can we try to model the spectroscopic signatures that could come from the X-ray experiment? So to do so, sort of like we collaborated with the group of Shaul Mukamel. In fact, we have DOE project covering 4 institutions. And there, our group has been providing the non-robotic dynamic simulations, whereas, for example, Schau's group was calculating all kinds of nonlinear signals. Here we are looking at the dimers that I've shown you before. And before, this dimer has been interrogated by two-dimensional spectroscopy in Greg Engel's group. And of course, sort of like there have been some signatures of the coherent vibronic dynamics identified. So it's known that we have a Frenkel-Light system, and between two monomers, we have appearance of the photoexcitation coherent dynamics. Now, what we would like to do here in terms of X-ray spectra, we would like to model the true car signal, that's an X-ray spectrum, where sort of like in a very simple term is analogous to the usual Raman spectroscopy. However, in the usual Raman spectroscopy, the vibration is a small energy, electronic transition is a large energy. And here we have electronic transition to be a small energy, whereas the core hole excitation would be a much larger energy. So there are sort of like a pump and probe. And what we would like to do is we'd like to use non-robotic dynamic simulations to provide all the input for modeling of those nonlinear signals. Let's go with it. So first of all, I'll just remind you, once you'll have this non-robotic dynamic simulations, then you have two states. And the dynamics between two states is coming through the regions of the low coupling when you have the localized wave function, and high coupling when you have the localized wave function. And of course, there are some vibrational degrees of freedom which are associated with these dynamics. Now, how can we calculate the true car signals? So in fact, the main ingredient to those simulations would be the time-dependent evolution of the entire wave function, having electronic and vibrational parts. And it is important to preserve all those phases. And by the way, sort of like I forgot to mention it, once you're doing sort of like ab initio multiple cloning dynamics, it is still trajectory-based approach. However, you run the ensemble of the trajectory, then you post-process all those trajectories using tabulated quantities, and you will recover the evolution of the entire electronic wave function, electron vibrational wave functions. Then by using this evolution, you are calculating the coherence between states. And in fact, we'll have the density matrix that Irene has mentioned. And one could look at the coherence as the evolution of the off-diagonal element of the density matrix, where all those coefficients like sort of like needs to be tabulated and post-processes, and they are defining so that the coherence would be governed both by electronic and vibrational degrees of freedom, so that I cannot put the label whether it's vibrational coherence or electronic coherence. And then once you'll have this, one could calculate sort of like the true car signal, which is essentially autocorrelation function of the wave function at the t equal to 0 and subsequent time. So that here we are realizing a full atomistic approach. We are starting from non-robotic dynamic simulation and going to the prototype of experimentally measured spectrum. So what we'll get? So let's take a look at the single trajectory example. So again, I'll show you later the other trajectories. So this one would be what is supposed to be a measured true car signals. And all those wiggles correspond to the coherences. This would be my populations. And you could see during the population analysis, we have crossing between those states, and those crossings are occurring intermittently as it would be expected. For single trajectory, we have a persistent coherence. And then sort of like you could see how the gap between those states behave. And then you could see how the electronic energy is going to the kinetic energy in terms of those crossings. Now, let's analyze how this coherence between those states appears. Let's look at the other trajectories. So this coherence can be either created upon photoexcitation, or it can be created due to non-robotic crossing between those states at the later time. So this is the case when the coherence is created upon photoexcitation, so that we have a strong coherent superposition between those states. And this persists until the conical intersection. So at the moment of the conical intersection, we have cloning event. Essentially, after this cloning event, the coherence is significantly subdued. In the opposite case, sometimes initially we have no coherence, and then once you go into the intersection, then this coherence would be created. And again, sort of like remember that those are the examples of the individual trajectories. And what we need to do is to analyze the signal on the ensemble of the trajectory. So you could see that in spite that we have the ensemble of molecular dynamics trajectories, which are starting from at room temperature at the very different conformational spaces, we still have a persistent coherence on the hundreds of femtosecond time scales. And during this time, we have a continuous crossing between the excited states. And of course, you could see sort of like with time, sort of like we have non-radiative relaxation to one of those Frenkel-Exciton states. So that I guess I'm running out of time. I'd like to leave some time for the discussion. Let me go into the conclusion by saying that currently non-robotic dynamics simulation are becoming useful, and Irene showed you a very beautiful examples how we can treat also realistic systems. Coherent vibronic dynamics is ubiquitous. And here we have seen that both electronic and vibrational degrees of freedom contribute to this dynamics. And intermolecular conical intersection essentially can be used to control and propel the energy transfer events. That's what I believe that people will be looking more and more in the solid states. And finally, the XFEL experiment can be sensitively probe coherences, and TrueCars is one of those experimental tools, experimental setups that can be used to probe those coherences. And of course, there are multiple people participating in the this study. Sort of like we have a large team, sort of like participating in development of the software for non-robotic dynamics. We have been collaborating closely with the theoretical team of Thomas Fraunheim, and Lynn Gross was a postdoc who did all those simulations of non-robotic dynamics to track the conical intersection. And we have been collaborating with Shal Mukamel on the TrueCar signals. And also sort of like I should give the credit to multiple experiments, because we are always relying on experimental work to guide our studies. And in particular, team of Christoph Lenau and Antonietta Desio from Oldenburg help us. We have been helping them to sort of like understand the dynamics of at the intermolecular conical intersections. And with that, thank you so much for your attention.
Okay, another very impressive theoretical talk. We'll just jump, we're almost at the end, but I'm going to jump into some questions. The first one is from our host. Do you see evidence of polaronic slowing of the exciton dynamics in your simulations or in your calculations?
All the time, sort of like, again, sort of like, we are not talking about polarons as the dynamics of a single particles. Mm-hmm. So the entire dynamics could be classified as evolution of the exciton polaron. Our exciton is always sort of like in the cloud of the vibrations. Mm-hmm. And those vibrations, and you could just point to the typical suspects, mostly that would be bond stretching and torsional motions. And those vibrations provide the polaronic effects for these dynamics and provide the best, effective best for relaxation of our excitation. And to me, sort of like in large molecules, the internal bath of vibration is the most immediate vibrational bath that is taking the excess of electronic energy.
Okay, another question. Physically, what motions constitute the GH coordinates, where G is tuning and H is the coupling coordinate? And I think this was around the middle of your talk. There was a like a two-dimensional.
So in a very simple approximation, so for the G and H, you have a dimer, and I sort of like I use the dimer model, if you wish, for both intermolecular conical intersection and for true Karsh signal. And then for this one, you have the bond stretching motion on each monomer. That would be one of those coordinates. And then you have the motion in between the molecules. That would be another coordinate. So again, sort of like, it's not exactly this one, but it can be sort of like wiggling, sort of like, but I would quantify those coordinates as intermolecular and intramolecular.
Okay. It's a simple distinction. Now, here's a question I also had, which is how does entry into a conical intersection create coherence? And I'll amplify, you're referring to electronic coherence, vibrational coherence, or both?
I believe I refer to both. So remember, sort of like, if you have the wave packet that is evolving on a single potential energy surface, and then it enters the conical intersection, and this wave packet is splitting into two wave packets. Then sort of like suddenly you have, originally you don't have any off-diagonal elements for the density matrix, and suddenly you have a split of the wave packet, and then the coherence is created.
Okay, and then that survives for just basically whatever the electronic dephasing time is, at least the electronic coherence term?
So we have seen it survives on the picosecond time scale, up to picosecond time scale.
Okay, in your simulations. And then one last question from the private chat. So you talk about intermolecular conical intersections, S1 to S1 sort of. What about conical intersections that couple the excitation to the ground state, like, like sort of traditional reactive type conical intersections? Do you see those as well?
Remember, sort of like, we are talking about photoluminescent polymers. In the photoluminescent polymers, there is a huge gap between the ground state and excited state, and this gap is over like 2 electron volts. So there is a lot of vibrational relaxation, and in fact, sort of like for the usual, I would say, photoexcitation in the weak field, unless you drive the system like crazy, there would be no conical intersections between ground and excited states. And this is a typical case. That's why those molecules luminesce. And also sort of like the relaxation to the ground state occurs on close to nanosecond timescales, which is a much longer timescale. So again, sort of like we don't see it. We have been looking at the immediate photoexcited dynamics on picosecond time scales of the closely spaced coupled electronic states.
Okay, but it could exist, I guess, in principle.
Well, again, could exist. So you have seen multiple examples from, for example, from DNA damage, from retinal. So once you'll have this strong photochemical dynamics, then that's it.
Okay, so maybe here, Sergei, I think maybe we'll end the official part of the workshop.